Pfiesteria has been in the news lately. Have you heard of Pfiesteria? I can't hear you. That's one thing you're going to find out is like I can't hear. No, it's not from not lack of hand washing. Is that? Hold on.
She said no
I guess that's not what she said.
She said no one watches the news.
It's not nice to make fun of handicapped people. Yeah, whatever. Okay, no one watches the news, right? This is the problem. This was the problem in the Bay. Under certain conditions, you had these little algal blooms, or you have this really high growth rate of this alga that causes skin irritation. It causes fish kills. I don't know what else. And what did you say back there? Okay, I heard that. Viruses. Viruses are in the field of virology. They are acellular, not cellular. They do not have any metabolic component to their life cycle. And since they are ametabolic, they are not metabolic. They require the metabolic functions of their host to carry out their life cycle. Okay, so what are the roles of microorganisms? We're talking about why microbiology is important. Okay, so or what are your interests in microbiology, and what are areas that you can explore in microbiology? So to answer that, we look at some of the roles of microorganisms, and we can look at recycling. Microorganisms have a really important role in recycling nutrients, so that when they are taken up and used to build macromolecules by one organism, the bacteria can break those down when they're no longer needed and recycle them into their subunits so they can be used again. So they are important in nitrogen cycling, in carbon cycling, oxygen and phosphorus cycling. Sewage treatment, indispensable for which is really kind of a part of this recycling topic. Sewage treatment is breaking down all of the waste products that are generated in a very populous area and rendering them safe. So recycling water is a very important role. Bioremediation, cleaning up pollutants, cleaning up toxic wastes, cleaning up oil spills. Oil spills are a natural phenomenon, just not on the scale that man creates them. Man can't make these huge oil spills, but eventually, all that oil that spills will be recycled eventually. Because you have spots on the ocean floor where oil seeps up naturally. And in those areas you can find bacteria that use that oil as a carbon source. All organisms need a carbon source. All of them do. What's our carbon source? Don't make me try to repeat something I can't hear, because it's embarrassing. Where do you get your carbon?
Is it air?
No. Are you a plant? That's what plants do. Sugar? Yeah, you get it from your diet. You get it from eating other organisms. Hetero, you're a heterotroph. Hetero means other, troph, eater. You eat others. Doesn't sound nice, but you do. And these bacteria get their carbon from oil. Plants get their carbon from the atmosphere. Insect control. Bacillus thuringiensis is a good example of a microorganism that is toxic, not toxic, that will kill, that's a parasite to certain insects. So that you can use it to control insect growth. Industry. The food industry, in particular, uses microorganisms to make cheese, other dairy products, vinegar, Beer, yes, beer and alcohol, sake, soy sauce, all of that stuff. In industry, it's important to control microorganisms. Control for food spoilage, control to prevent food spoilage, to prevent poisoning. Genetic engineering, getting bacteria to make products that you can use as drugs. Genetic engineering is using the gene from one organism, introducing it into another organism so that other organism will make the product of that gene. All sorts of different manipulations you can do with that. Proteins are made, such as enzymes, insulin, growth hormone, interferon. All of these things can be made by genetic engineering. Oh, and vaccines as well. Gene therapy. Gene therapy is taking a gene that codes for a protein, and instead of making that protein in another organism in the laboratory and then taking the product and then giving it to whoever needs it, is to take that gene and give it into the individual directly and let the recipient body express that gene. That's what gene therapy is. You can do this in animals and you can do it in plants. What are the roles of the normal flora in the body? The bacteria that are present in your body normally have a protective role. And they also have a nutrient type of role. Some bacteria produce vitamins for you to use. Some bacteria keep other harmful bacteria out, and they're just kind of living there. And it's okay if they stay there because they keep out the bad bacteria. So what's the normal flora role? And disease, when those normal flora get out of hand or if they're unbalanced. You seen those Nutria commercials? Nutria? No. Neutrogena? No. It's a woman selling the yogurt. Activity. Yes. Well, that's kind of, I don't know if that really works, but that's the idea. Okay? The good bacteria will crowd out the bad bacteria. So when that balance gets upset, you got to replace those good bacteria. So the disease-causing bacteria don't take hold, or you can get rid of the disease bacteria sooner by introducing the good ones. Emerging diseases or re-emerging diseases. Emerging diseases are new diseases in an environment that are brought about by different disturbances. Sometimes some people think, deforestation is a cause of some new emerging diseases. Or diseases that have been kept in check historically are now finding their way back, like TB, like what else? Pox, polio, smallpox, polio. Not in the United States, but in other areas. So emerging diseases or re-emerging infectious diseases, they have a significant impact on populations. Oh, here's a new one, an emerging disease, the flesh-eating Streptococcus pyogenes, the flesh-eating disease.
Is that what we just saw?
Yeah. Right, yeah. That's relatively recent. Oh, there it is. Causing that necrotizing fasciitis. Oh, and what is the role of antibiotic resistance to re-emerging diseases? So bacteria that have been always kept in check with antibiotics, or at least since the mid-40s, are now becoming a problem again because they are resistant to drugs.
Do you mean like MRSA?
Yes. Methicillin-resistant Staph aureus. Or hemorrhagic fever, Ebola virus. That's a relatively new disease. Listeria, new disease. This is the one that I couldn't hear. So we're talking about what are the reasons for these reemergent diseases. Well, there's genetic components to this thing, genetic being like changes in resistance, drug resistance. And evolutionary changes, how the bacteria themselves change their genome just through natural evolution. Become a problem where they weren't a problem before. It's supposed to be the other side around. Just turn upside down, you'll see it's in the right direction. But, modern transportation spreads diseases where they haven't been spread before.
Have you seen that movie? Contagion? Oh, no.
Yes, terrible. Deforestation, construction, kind of talked about that. Okay, history. Oh, what time is it now?
7:38.
Oh, very good, thank you. How did I miss that? Okay, history. Tools allow you to do new things. So the microscope is really the first tool that opened up the whole world of microbiology, because now people could see that there's something else in that makes up their world. There's something else. There's a component that was undescribed before this time. So the microscope invented, the first microscope by van Leeuwenhoek, showed that there was simple microorganisms and that larger organisms were made up of smaller cells. So there was a cellular-based tissue, and that there were single cells organisms in the environment. Do you know this kind of, okay, do you know where we found these kind of single cells? No. Interesting thought. No. And he looked at a lot of different things, but one of the things he looked at was scrapings from his teeth. So think about it. These people never brush their teeth. God, it really must smell. So yeah, beginning to see living organisms that were unknown at the time. We can kind of talk about this now and then later too, but so you see something new, right? And you keep watching it. And you see what it does, and then you try to make a prediction about it. This is the scientific process, the scientific method. And the scientific method was key in developing the science of microbiology and all science, because it was a systematic way, or a systematic approach to learning something new. So you have these observations, and then you try to make a guess, what to explain the observations. And we call that a testable question. You come up with a hypothesis. A hypothesis is a statement. Okay. Crickets will jump when I see, I don't know, that's not a good one. Let's see. I don't know. Here's a stupid one. Crickets jump when you make a loud noise. Okay, you see crickets jumping, but you only see them jumping when there's some kind of noise going on. So you say that crickets jump when you make a loud noise. So you could say your hypothesis could be crickets have good ears, which allow them to jump. Something like that. Okay. So then you have to test it. Then you come up, that's the experiment. You come up with an experiment to test your hypothesis. How do you test the ear thing? Well, You remove their ears and see if they jump. So you don't know where the ears are, so you take off the legs. Oh, they don't jump anymore. You make a loud noise, they don't jump because you took off the legs. You're assuming, of course, that the ears are in the legs. So at the end of that experiment, would you accept or reject your hypothesis? Did they jump when you made the noise? No. So you have to accept your hypothesis until someone else comes around and comes up with a different way to test it, or a different hypothesis, a different way to test the same hypothesis. And then you can, at the end of every experiment, you accept or you reject the hypothesis. So what does the scientific method do? It gives you an approach to disprove things. It doesn't prove them. It only disproves. So that's what the scientific method is. You have an observation, you come up with a hypothesis, you come up with an experiment to test the hypothesis, and then you accept or reject the hypothesis. And so this is how scientific progress has been made, by using these simple steps. Early on in microbiology, in the history of microbiology, there were two different theories that were competing. One was biogenesis, the theory that living things gave rise to other living things, and that's the only place you got living things was from other living things. Or spontaneous generation, that says that non-living things come from, that living things come from non-living things. So the meat that's rotting gives rise to maggots. Okay, so the flies came from the dead meat. But we now know, of course, that the flies came from eggs that were laid on the dead meat, right? So you come up with a method to test your hypothesis, and that's how progress was made. Disproving spontaneous generation. You know, people came up with these hypotheses, they tested them, they came up with a conclusion, which made them reject the idea of spontaneous generation. And here's the experiment that Pasteur did. He had these what he called swan neck flasks, which are flasks that have a very thin opening, a narrow neck opening. He filled them with growth medium, like a soup, that was boiled to kill all the microorganisms. And he put an S-shaped curve into the neck of the flask. So he compared this flask to a flask like this, where the neck was straight up and open. And the hypothesis was that bacteria are contaminating the soup, making it grow, making it spoil. So he came up with this experiment to test that hypothesis. So this neck stayed open, straight up to the air. This neck was bent. Although it had access to air, there wasn't a direct route. So the bacteria couldn't go in here, according to his hypothesis, couldn't go in here and climb up the wall and then in. They could only get in if they dropped straight in. So they were both open to air. And lo and behold, I don't have the results. This is the only one here that grew bacteria of the two in the comparison. This one stayed sterile, which supported his hypothesis that the bacteria in the air were causing the spoilage. Did it prove it? No, but it hasn't been disproven yet, and that was a long time ago. And since then, there's been other variations of this experiment. Sometimes air was pumped through with bacteria the air was pumped through, and then it would contaminate, but then the In one experiment, the neck wasn't heated. In another experiment, the neck was heated so that the airplane pumped through with bacteria. The heat would kill the bacteria, so by the time the air got into the flask, it was sterile. It's just another way to test the experiment, another way to test the hypothesis. Okay, so in this method, biogenesis became more favorable in public opinion. I talked about that already. Okay, theory of biogenesis. Living cells come only from other living cells. Pasteur in the mid-1800s contributed a lot to this work. His work with fermentation. So he knew that fermentation, making alcohol, was a microbial process, and he controlled it. He came up with the technique of pasteurization, which is a way to stop microbial fermentation or to stop the spoilage process in liquids such as milk. What's the deal about pasteurization? How does that work to stop spoilage of milk? It's a process that he invented.
The bacteria?
Did they put bacteria in the milk?
No. They're killing the bacteria in the milk.
Bacteria, because the bacteria can only stand up and resist it, so it's certain amount of heat.
Right. So he would apply heat to kill the bacteria in the milk to preserve the milk. Why don't you just boil the heck out of it?
It would ruin the taste, or it would do something to the taste.
Yeah, it wouldn't taste as good. Boiled milk doesn't taste very good. So the fermentation process rapidly increases the temperature to like 80 degrees and then drops it very quickly. And then does it again and drops it really quickly. So that way you preserve the proteins in the milk without destroying them. You preserve the flavor, but you kill the bacteria by these successive heating and cooling periods. He also developed aseptic technique. Aseptic technique is a method that you use in the laboratory to keep bacteria out or bacteria in of whatever you're doing. So if you have something that you want to keep sterile, it's a method to use to keep it sterile while opening it to the environment. So if you want to study bacteria in pure culture, you're going to have to keep out other bacteria, right? Because you don't want them contaminating your culture, otherwise your experiments or your identification process is going to be all messed up. So you use these techniques to keep the bad bacteria or the unwanted bacteria out and your bacteria in. And it's a method of heating glassware, sterilizing the tips that you're using to inoculate. You know the technique. I just showed you one little technique in the lab today. What was that? That has to do with good aseptic technique. Yeah, that's part of it. That's not what I was thinking. That's a good one. You know, the way you take off the cap of the tube. There's another part of that I didn't show you, and that's heating the lip of the glass to kill any of the bacteria. We're going to go over that in the lab. And then you put that cap back on without touching it. What was another method that was very subtle?
How you open the swab.
The way you open the swab. Okay, that's not what I was thinking, but that's good.
How you open it on the auger.
Yeah, the way you open that Petri dish, right? Upside down. Keep it upside down so stuff doesn't fall. That's aseptic technique. It's a collection of techniques that we use in the laboratory. That's my own personal one, by the way. So Pasteur developed the germ theory of fermentation. He studied fermentation and realized that it was a microbial process. Pasteurization, which we already talked about. What's the germ theory of disease? Easy. You can make it up. You would use most of those words in your answer. Germs cause the disease. Very good. Yeah, germs are the cause of the disease. What's the rationale behind the germ theory of disease? And that's something you might not be so apparent. Why did people think this? What was going on during this day, during this period?
It's like
The plague.
Huh?
The plague.
No, we haven't even talked about the plague yet. I'm talking about, people having a good time inoculating fermentation drinks and drinking them and having a good time, realizing it's a bacterial process. Well, they thought that, well, since this fermentation thing and the spoilage thing and the pasteurization thing is all centered around bacteria, right? All centered around bacteria. Well, maybe disease is caused by bacteria too. So that's when they started looking into other causes for disease. That's the rationale behind the what was it? the bacterial cause of
Theory of germs.
the what?
Theory of germs.
Yeah, germ theory of disease. Thank you. So I got home. And that's it. We'll pick up there next time.